Okay guys, so last time we talked about the central dogma of biology, and that is the information flow goes from DNA into RNA into proteins. And is DNA a macromolecule or a micromolecule? Is DNA a macromolecule or micromolecule? Micro? Is DNA a micro or a macromolecule? It's a macromolecule. It is made up of nucleotides. So the building blocks of proteins are amino acids. The building blocks of carbohydrates are monosaccharides, disaccharides that will give us polysaccharides. No, you guys are mixing things up. Okay, how many macromolecules do we have? How many macromolecules do we have?
4.
We have 4. The first one is your proteins. So we have proteins, and these proteins are made up of the amino acids. Excellent. Amino acids. So proteins are made up of amino acids. And amino acids look like this. You have an alpha carbon, and it has 4 terminals or 4 substituents. The alpha carbon has a hydrogen group, and it also has a carbonyl carbon to whom OH is added, and it has an amino terminal with 3 H's. And then what makes amino acid 1 different from amino acid 2 is the R group. The R group is what makes the amino acid different. Then if you have another amino acid, it will also have an alpha carbon with a hydrogen terminal with a carbonyl carbon, and you have an OH. On the amino terminal you will have N with 3 H's. And what's going to make this different from, what's going to make this amino acid number 2 different from amino acid number 1 is the R group. The R group is what makes the amino acid different. So if I want to, so these are the micromolecules. The amino acids are the micromolecules. These are your micromolecules. The proteins are known as macromolecules. So we go from, there is a bidirectional relationship. If you go from proteins, if you break down from proteins down to amino acids, if you break down from proteins to amino acids, what is that known as? If you break down from macromolecules to micromolecules, from proteins to amino acids, what's the name of this process called?
Reduction.
So that's catabolism. So we begin very big. So very nicely, your classmate said hydrolysis. That's true. But now we're talking about how it happens. So we need to begin very big. That's catabolism. When we go down this road, that's catabolism. That's breakdown. We are breaking down. That is breakdown. And the breakdown happens with water. Hydrolysis. Hydrolysis. That is hydrolysis. And if you want to build up, then you go the opposite direction, and that is known as anabolism. That is anabolism. Anabolism is the build up. It is the build up of macromolecules from micromolecules. And for that we use dehydration. Excellent. We use dehydration, also known as synthesis. So you have these 2 amino acids. If I want to join them up, I utilize this very important bond. I utilize this very important bond right here. You see where the green marker is. This bond. So what happens is I have an OH here and an H here. If I remove that, then I go up. I build proteins. If I remove that, I minus water. I go minus water. I take away water, minus H2O. I remove the water. So water came out. So the arrow is pointing outward. The arrow is pointing to the outside. If the arrow points outside, I subtract and I took away water. When I take away water, now both of them are going to join. So now I'm going to have my same amino acid. It has, say with me, that's the alpha carbon. And it has 4 terminals. One terminal is hydrogen. One terminal is the alpha carbonyl because it's attached to the alpha carbon with an OH. And on the amino terminal, you will have N with 3 H's. N with 3 H's. And the only difference is the R group of 1. That's what makes it different. That's the R group for 1. And we have our second amino acid that looks like an alpha carbon with a alpha carbonyl. That's an alpha carbonyl that is attached to the alpha carbon with an OH, and it has a hydrogen terminal and an amino terminal and N with 3 H's. And I need to put an R group. I need to put an R. That's an R1. So now these 2 amino acids, I build them up by taking out water. So if the water is gone, that means OHs are gone. The OHs are gone. The O and 2 H's are gone. So now water is out. And instead, I have this very important bond. What's the name of this very important bond? That's a peptide bond. That's a peptide bond. That's what I'm building up. But if I want to break down, when I want to go down, what happens when I point in this opposite direction? Now I add water. If I want to split, I add plus H2O. I add water, and when I add water, the water has O with 2 H's. If you add that in there, you're going to separate it and cleave it into an OH on one side and an H on the other side. Okay? So you have to know that. You have to know that we break down, that's catabolism. We build up, that's anabolism. When we break down, do we get energy? Do we get energy or do we lose energy when we break down? When we break down, do we get energy or do we lose energy? So when we break down, we get energy. That is known as exergonic or endergonic. If you gain energy, that's exergonic. Excellent. That is exergonic. Exergonic reaction with a positive delta G, exergonic reaction. But when I'm storing, I'm not breaking down anymore. What is that called? So now when I'm building up, that is known as endergonic. And that's an endergonic reaction with a negative delta G. We mentioned exothermic and endothermic. Endothermic, therm means temperature. So endothermic is when I add energy to the bond. You see these bonds? They're full of energy. When we see fat in a little bit, how do we get energy from proteins? How do we get energy from carbohydrates? How do we get energy from lipids? By breaking them. When we break them down, they liberate energy. It's like when you squeeze a lemon, the lemon juice comes out. So you break this bond, then you get energy. So we catabolize and we use this heat energy to break down, but we get free energy. But when we are building up, we consume energy to build up these molecules. So metabolism is the sum of both. So anabolism plus catabolism, that will add up to metabolism. And metabolism is the sum of all the biochemical reactions that are concerned with storing energy in anabolism. Gaining energy by breaking down all of the macromolecules. So you have to know each micromolecule, how do we assemble them? And when we assemble them, do we consume energy or do we get energy? So when do we consume energy and when do we get energy? Do we consume energy in catabolism or do we get energy in catabolism? We get energy. That's exergonic. So the energy exited the bond and came to me. That's exergonic. And what happens when I add, like when I'm assembling, and now I am endergonic, what happens? I'm not gaining energy. I am investing my energy into the bond, into building up this protein. So that's all about proteins. But we are not done. So our proteins are very useful macromolecules, and we talked about what is their usefulness. We said that proteins are enzymes, and when we talked about the cell, the cell has red heads. Okay, so in order to talk about the phospholipid bilayer, we got to know what a lipid is, and we got to know what is a sugar. So let's move on now from proteins to lipids. Does anybody have a question about proteins? Nobody has a question about proteins. So now let's move into lipids. You have the same way we had two arrows of micromolecules and macromolecules. We're also going to have two arrows for lipids. So okay, that's macro. I can't even show. We have macro molecules, and on the bottom we have micro molecule. So what is the building block of lipids? So the macromolecule is going to be a lipid. How are we going to get a lipid? We're going to get a lipid by joining two macromolecules. And these are your glycerol backbone, which is 3 carbons with OH. That's glycerol. You need to know that. That is glycerol. Glycerol is going to bond with 3 fatty acids. Carbon. That's a carbon. That's a carbon. And then okay, so that's the glycerol backbone. What is that? That's the glycerol backbone. What is that? That's the free fatty acid. It's free. That's free fatty acid. Okay, that's number 1. That's the second one. Free fatty acid. and that is a free fatty acid. Those free fatty acids now are going to assemble together. So when they assemble, what's going to happen? Look at this. You still have an H and OHs. H and OH. H and OH. So what do you think is going to happen here? What do you think is going to happen? If I want to assemble, I want to make a big, these are two separate micromolecules, or four actually, four. Two of the same two of different kinds, but one of the two is there's three of them. So what is going to happen? We're going to remove water. So we take out, the arrow is going to point out, we take out minus H2O, and then I'm going to end up with a glycerol backbone. First carbon, second carbon, third carbon, okay? And I'm going to have a bond, and that is going to be my carbonyl, carbon, carbonyl, carbon, carbonyl, carbon, and attached to all of these carbons, all of these carbons, and the bond is going to be right here in the middle between the carbonyl and the carbon, carbonyl and the carbon, carbonyl and the carbon. What is that now called? What is that now called? That's a because there's three of them. Triglyceride or triacyl glycerol. That's a triacyl glycerol. Triacyl glycerol or triglyceride. So you need to know that. You need to know what glycerol is. You need to know all of these free fatty acids and how we assemble them and how we break them apart. So now we have assembled them, and something very interesting about the phospholipid bilayer. How does the phospholipid bilayer look like? It looks just like that. You have your three carbons, okay, and two of them are attached to a carbonyl, and then you have carbon tail, carbonyl, and carbon tail, carbonyl, and carbon tail. But does your plasma membrane look like that? Does your plasma membrane look like a triglycerol? It does not. So what's different? One of those carbons does not have a hydrophobic tail. It has a big red P. It has a big red negatively charged P, and on top of it there is a group known as choline. This choline. So now that's what makes the hydrophilic head. It's this red phosphate group, and the two are the tail. So your phospholipid bilayer, hydrophilic heads, and hydrophobic tails, they are actually a triglyceride that lost one tail and substituted that lost one tail with this phosphate and choline. That's what makes the head hydrophilic. So our plasma membrane looks like that. Our plasma membrane looks like this. It has hydrophilic heads dissolving in water. Hydrophilic heads dissolve in water. You see the hydrophilic heads dissolve in water, and it's a bilayer leaflet. So that means the top have hydrophilic heads. The top have hydrophilic heads, and the bottom have hydrophilic heads. And in the back you have a very important tail. You see, these are hydrocarbon tails. These are the hydrocarbon tails. These are the hydrocarbon tails. These are the hydrocarbon tails.
Professor?
Yes.
Question.
Yes.
You saying that phosphate it attaches to the where do those two things come from, the phosphate and the choline?
That's just how they are synthesized. They are created like that. The cell, there's enzymes that facilitate the creation. So it's like a factory. That's how it's made. So there's enzymes that remove, cleave this bond and assemble a phosphate and a choline to prepare it to become part of our plasma membrane. That's how our plasma membrane is. Okay, so you just need to know that your plasma membrane has two hydrophobic tails and one head that's phosphate and a choline. That's the red one. And in the core, right here, in this core, you have a butter. All of this is butter. This is hydrophobic. So that is hydrophobic right here. All of this is hydrophobic. That's hydrophobic. And the top here, that's known as hydrophilic. You get it? That's hydrophobic and that's hydrophilic. Now, these hydrophilic heads are red. Red because why are they hydrophilic? Why are they red? Why are they hydrophilic and red? Why are they hydrophilic?
Because of that phosphate.
Because of the phosphate. And why is the phosphate making it? Why is the phosphate making it hydrophilic?
Because of the choline.
Because it's what?
Of the choline.
No, it's not because the choline has a nitrogen. The choline has a nitrogen, but why does the phosphate make it polar? Because it's charged with a negative charge. You see this negative charge? Don't forget this negative charge. The negative charge, that's why sodium chloride dissolves in water, because sodium is positively charged and chloride is negatively charged. Okay? So this phosphate negative is what makes the polar head red to dissolve in water. Okay? So you have to know that. You have to know that this trilipid, for the exam, this trilipid glyceride look like that. Right now, when you look at it like that, is it nonpolar? Is it nonpolar?
Yes.
Yes, it's nonpolar. Is it lipophilic? Is it lipophilic?
No.
Lipophilic. Philic means love. Lipolipid. Does it love lipids?
Yes.
Yes, it's lipophilic. If you're nonpolar, you're immediately lipophilic. If you are nonpolar, it means you do not dissolve in water. So you will dissolve in oil, you will dissolve in lipids and detergent. That makes you immediately lipophilic. That makes you lipophilic. But if you are polar, that makes you lipophobic. And lipophobic means hydrophilic. Lipophobic means hydrophilic polar. Non-polar means lipophobic and hydrophobic. Okay? This is not new. Polar means you have a charge. You can dissolve in water. You're hydrophilic. So that means you can never be lipophilic. You're lipophobic. Okay? That's very important. Okay? I'm going to post this up for you so you don't have to take photos. I'm going to post it. But if you want to, you can, but I'm going to post it up for you. And attach it in the announcement. Okay, that's nonpolar, lipophilic, hydrophobic. Polar, lipophobic, and hydrophilic. Okay, so right now, when you look at a triglyceride, is it nonpolar or polar? It's nonpolar. You see, because it has all carbons. Okay, it cannot dissolve in water. But what about the phospholipid? When I did a modification, now I did a modification. I removed one whole free fatty acid and instead put what? What did I put instead of that one whole free fatty acid? What did I put? Phosphate only? Phosphate and choline. The choline has a nitrogen and the phosphate has a minus group. This phosphate is the red part. And that's why 2 tails, 1 phosphate head. 2 tails, 1 phosphate head. Okay? Are you okay? Alright. Okay, are you guys following? We have 2 tails and 1 phosphate head. 2 tails, 1 phosphate head. 2 tails, 1 phosphate head. Okay? Alright, now we looked at if we want to assemble, we want to go from micro to macro, from free fatty acid to triglyceride. We join them by minusing water. Water is gone. We subtract water. Water left. But let's say I want to break down. If I want to break down, then I add water. So I add plus H2O. When I add water, then the OH is going to go to the glycerol and the H is going to go to the free fatty acid. Is that clear? Then we talked about this membrane. It has different kinds of proteins. It has a protein that stay only on the outside, on the periphery. You know we have 2 leaflets, 1 leaflet at the top, 1 leaflet at the bottom. That's no longer a secret. We have 2 leaflets, 1 at the top, 1 at the bottom. That's the core of the cell. That's the cytosol. And that is the extracellular environment. Are we following? That's the extracellular environment, and that is the intracellular environment. So that's a peripheral protein on the outer leaflet. You guys see the peripheral protein on the outer leaflet? There is another peripheral protein on the inner leaflet. That's a peripheral protein on the inner leaflet. That is the peripheral protein on the inner leaflet. Okay? Then we have another kind. This other kind of proteins is a transmembrane. So that's known as the integral protein because it spans both layers. It spans both layers. You see? It's in both. Inside and outside. Inside and outside. It's both. Scanning both. Spanning both. So that makes it, these guys are known as peripheral. Peripheral proteins on the outside. And these are integral proteins. Integral proteins. These integral proteins are transmembrane. Transmembrane proteins. These are transmembrane proteins, integral proteins. So being peripheral means you are on 1 leaflet, on the outside of 1 leaflet, or on the bottom of a 2nd leaflet. An example of the integral protein is what? Is a receptor. You know, we talked in the past, we said inside our nucleus, inside our nucleus, we have our DNA. We have our DNA. When you transcribe the DNA, this nucleus has an envelope, and the envelope has holes, like a membrane. All of the organelles of the cell have holes. All the organelles of the cell have a hole. The Golgi body have a membrane. The endoplasmic reticulum has a membrane. The ribosome has a membrane. Every single organelle in the cell has a membrane. So what happens now, this nucleus has a membrane. You guys see the membrane of the nucleus? Can you guys see the membrane that I'm drawing here? Can you guys see? Can you guys see the nucleus and the membrane that has holes? What happens now is if you have, this is very important, if you have a steroid hormone, are we done with that? Are we done with, did you write this? Did you write that triacylglycerol, we remove 1, we put a phosphate and a choline, and then what makes the hydrophilic head and the tails are the 2 ones that are in the back? Are you guys done with that? Okay, so now let's look at what happens. if we have two different classes of hormones. We have two different classes of hormones. These two different classes are classified structurally based on their structure and solubility in water. So there are hormones that dissolve in water. We have hormones are divided into two classes based on what? Based on structure. So one that dissolve in water, that makes them say with me, that makes them hydrophilic. And that makes them polar or non-polar? Polar. And does that make them lipophilic or lipophobic? These are lipophobic. And they are known as catecholamines. Catecholamines. These are catecholamines. And then there are hormones that do not dissolve in water. They don't dissolve in water. And these are known as steroid hormones. Steroid hormone. All of them, they are made up from steroid. If you have dough, butter, milk, you can make pancakes, you can make bread, you can make cakes, but the raw material is the same. All the steroid hormones are made from cholesterol. They're all made from cholesterol. These are known as cholesterol-based hormones. Steroid hormones are cholesterol-based hormones. Cholesterol-based hormones. Cholesterol-based hormones. These cholesterol-based hormones are made from cholesterol, such as aldosterone. That's your aldosterone, progesterone, estrogen, testosterone. The guys love this one. They get it from the exogenous. We're going to talk about that when we're talking about the endocrine system. You know, testosterone has to be synthesized from the testes, not to be synthesized, not to be taken as an injection. So, because they are young, and the guys in the gym, the bodybuilder, they are young, and they are enraged, and they want very quick results, they take exogenous testosterone. This exogenous testosterone is going to do bi-loop inhibition. It's going to inhibit the hypothalamus from secreting GnRH, and it's going to inhibit the pituitary from releasing FSH and LH. We will talk about that. So what happens now, there's a lot of testosterone in the body, so there will be feedback, negative feedback. Your testes are going to say, there's no need for me. There's already testosterone in the blood. So they're going to stop making testosterone. And their testicles are going to get so tiny and so small because they are not working. Because they take the exogenous. We're supposed to have endogenous testosterone. So this testosterone, progesterone is all based on steroid hormone and is based on cholesterol. So cholesterol, testosterone, we make testosterone from cholesterol. We make estrogen from cholesterol. Females also, because some females, they cannot accept menopause, so they go and they do like estrogen replacement therapy, and they do so much estrogen that it causes breast cancer. So these hormones, they are very dangerous, but when we get to them, we will understand them in depth. So these hormones that are steroid based, they are always made from what? Cholesterol. So now, based on this information, if I have a steroid hormone like testosterone, aldosterone, progesterone, testosterone, estrogen, these guys, can they penetrate into the cell? Can these guys who are steroid, cholesterol, non-polar, they are non-polar, They're non-polar. Can they enter the cell? What do you have here? What do you have here? You have what do you have here? In the core. What do you have in the core here? Brown. What is brown? What is brown? Brown is non-polar. That is non-polar. That is lipophilic. That is hydrophobic. That is hydrophobic. And the red is hydrophilic. Hydrophilic. So now these guys, they're hydrophobic, they're non-polar. Can they go through this butter, through this brown? Yes. Because like dissolves like. Because butter dissolves in oil. But oil cannot dissolve in water. Because water is polar, oil is non-polar. So here you have a non-polar core. So I know some of you may think, but I have a polar head. Can that? Yes, they can. They can zigzag through the polar head. It is this big guy who will block them. But if you are a steroid, non-polar, it can go through. Do you guys understand? If you're non-polar, if you are a steroid, you can go through easy. The steroid hormones, they go through easily, zigzag, and actually they can bind receptors that are on the cytosol. This is the cytosol. That's the cytosol receptor, okay? They can bind to receptor on the cytosol. They can even go and bind receptors on the chromosome. And if they bind on the outside, then there will be translocation. So now this is known as the mobile receptor. And this mobile receptor is going to mobilize when it binds the testosterone. This is my testosterone. This is my testosterone or cholesterol-based hormone. Testosterone. Or any, we'll just call it steroid hormone. I don't want you to think that this only happens with testosterone, estrogen, progesterone. So they go through the membrane, they're welcome. They have the ID. They can dissolve in the bilipid layer. And then they bind to, some of them bind receptor on the cytosol, on the cytoplasm. Some of them bind receptors on the chromosome. If you are on the outside, you bind on the outside, and then the receptor is going to mobilize. It's going to start swimming till it goes inside and then bind the chromosome. Or you can go already inside. They can go through two membranes, the membrane of the cell, membrane of the nucleus, and then they will do what? Transcription. They will transcribe. So you know we have, when you go in the nucleus, you have transcription. This transcription happens inside the nucleus. So we transcribe what? What do we transcribe? What do we transcribe in the nucleus? DNA. We transcribe the DNA to get what? RNA or mRNA. RNA is many types. There is rRNA, tRNA, mRNA, microRNA, small nuclear RNA, snRNA. There is many different RNAs. What RNA are you talking about? Yes. So that's the central dogma of biology. We go from DNA to mRNA through transcription. We go from mRNA into proteins through translation. This transcription happens inside the nucleus, and this translation happen inside the cytoplasm. Are we clear? So now we transcribe an mRNA. The mRNA now will go out. The mRNA will go out and it will be translated in the ribosome. So the ribosome is going to translate for us There will be translation. The translation is always outside the nucleus, and the transcription is always inside the nucleus. Then we're going to translate, then we're going to get our target protein. Our target protein. And then our target protein is going to give us what? It's going to give us a target response. We want a target response. That is the point we want, a target response. We want a target response. That target response could be to initiate catabolism. That target response could be to make more protein channels, block them, put them in the plasma membrane. So we got our target response. But how do we get our target response? This is our testosterone. That's our hormone known as the first messenger. That's the first messenger. The first messenger penetrated all through. The first messenger It's not working. Okay. So the first messenger made it all the way through. But now look, if the first messenger now is a catecholamine, like epinephrine. Epinephrine. Why do we need epinephrine? Epinephrine is released, it's like adrenaline. It's released in the times of stress, when you are stressed out. So it can give you more energy. So you can run away from the lion. Okay? So you can deal with a stressful situation. So now epinephrine is not like these guys. It's not steroid based. Epinephrine is not hydrophobic. Epinephrine is not hydrophobic. Epinephrine is not lipophilic. Epinephrine is polar. It dissolves in water. Now can it go through? Can it cannot go through, definitely cannot. It will be blocked by who? Good.
It's the tails.
The tails. This core, this brown, is going to block it. That is going to block the norepinephrine. So now what's going to happen? Norepinephrine is not going to work? It will. There's a peripheral protein here who will bind norepinephrine. Norepinephrine will bind outside here, and that is known as that's our first messenger. And it's our ligand. And it will bind the outside receptor, and that will cause a conformational change. It cannot enter, it can only bind from outside. It can only bind from outside. Then it will cause a conformational change, mechanical change in the receptor, in the receptor. And then the receptor is going to undergo this conformational change, and the conformational change is going to initiate another peripheral, so a peripheral protein outside binded the ligand. Then a conformational change, mechanical vibration, loop, like an engine started working. When it started working, another peripheral protein here known as G protein. This G protein is going to go, this G protein is going to go, and it's going to go to a very important protein here known as adenylate cyclase. It's another transmembrane protein here known as adenylate cyclase. Cyclase is a very important word because it's going to take ATP, adenine triphosphate, ATP, and convert it into AMP, adenine monophosphate. Adenine monophosphate. This adenine monophosphate is going to be cyclic. We put a C in there. Cyclic AMP. Cyclic AMP is our second messenger. That's our second messenger. That's our second messenger. Yes?
Could you please explain the first message?
So the first messenger is outside. It cannot enter. So on its behalf, somebody who's in is going to get the signal from the receptor. The receptor will say, oh, I detected norepinephrine, but he cannot get in. So there will be a conformational, mechanical, vibration change that happens in this receptor, and then the receptor is going to activate a G-link protein. The G protein is going to go activate adenyl cyclase. Adenyl cyclase is going to take ATP, convert it into cyclic AMP. Then cyclic AMP is going to go inside the cell, and cyclic AMP is going to activate kinases. Kinases. What are kinases? You're going to see this a lot. You're going to see the kinases a lot from this point onward when we're doing anatomy and physiology forever. This kinase is an enzyme that will add a phosphate group. It will do plus PI. It will add a phosphate group. Some enzymes, when you tag them with phosphate, they will start working. You will activate them. Other enzymes, when you tag them with phosphate, they will stop working. But kinase doesn't care. If you start kinase, it's going to add a plus phosphate. So the second messenger is going to activate a kinase. The kinase is going to go to an enzyme and add a phosphate to it, tag it with a phosphate, put a sticker phosphate. When you add a phosphate to it, that enzyme now is going to give us the target response or activate another enzyme that will give us the target response. So here, we're going to activate enzyme. So we went from off to on, and then this activated enzyme will give me a target response. So, yes?
So, does the epinephrine ever get in?
Of course not. That's why we have all of this happening down here. Because it cannot get in. We have all of this mediating the effect for it on its behalf. Like somebody cannot get in the school.
All of this is meant for the epinephrine to not get in. All of this.
Yeah, you can do that. I like that. Yeah.
All of this happens so that
That's what I was asking before. Does the epinephrine ever get in? At
Okay.
what point does it get in?
Okay, let me ask you a question. Let me answer your question with a discussion question.
Yeah.
Let's look at testosterone. Testosterone got in. Testosterone or its friends, those who can get in, bind a receptor and transcribe, give me mRNA. The mRNA give me a protein. The protein give me the target response. Norepinephrine, if it can get in, it can just go in and do that. But the target effect is something else. The norepinephrine can never get in. But once it binds on the outside, all these guys do that kind for him, to mediate his effect. It's like somebody
Okay, I get it.
can get inside the cell, inside the school, and start the fire alarm. That's like testosterone. He has the ID, he can go in. But somebody can be on the outside and say, please ring the bell for fire alarm. Then the security is going to go up the first floor. Tell the secretary. The secretary is going to go to the administration. The administration is going to contact the custodial staff. The custodial staff is going to go and ring the bell. So the issue here is you needed a second messenger. That's a second messenger because the first messenger cannot get in. Never. So this is what I want you to understand. For now, you have to understand that we have two systems. Even though both of them are first messengers, do not think that epinephrine is the second messenger. It needs the second messenger, yes. But it doesn't mean it's the second messenger itself. If you are on the outside, if you have a job to do, that makes you a first messenger. These guys are lucky. The steroid hormones, they are made up of lipid. They can go through easily, no problem. But it is those who cannot dissolve, those who are non-polar, they can never get in. So they bind on the outside and on their behalf, these guys, the G protein, the receptor is going to do a conformational change. The G protein is going to elicit adenyl cyclase. Then adenyl cyclase is going to have cyclic AMP. That's the second messenger. Then cyclic AMP is going to activate a kinase. Then the kinase is going to stick a phosphate group on the enzyme. Then the enzyme that had a phosphate group is going to be activated. Then the activation is going to lead to the target response. In the end, we want a target response. But how do we get it? That's what's different. If you are non-polar, you don't need a second messenger on your behalf to mediate anything for you. You can do it yourself. Go all the way. In the nucleus, like I said, some of them go to the nucleus. Others, they go to the cytoplasm and then mobilize to the nucleus. But then others who are polar, others who dissolve in water.
That's a facilitator.
Yeah, but we use the word, that's a dangerous word, facilitator. We use that for the protein channel. So now when you use the word facilitator, you're going to think of the facilitated diffusion. So facilitated diffusion, you're going to need a membrane channel that will facilitate the movement of glucose down concentration gradient. Make sense?
Yeah.
But those guys, we don't call them facilitators, we call them mediators. Mediators, they mediate. Okay? So that word facilitator is reserved for the glucose. But one of the target responses, I said already that one of the target responses is to make facilitated proteins. So for example, if there is so much glucose outside, a hormone can cause the transcription and making of proteins, and these proteins are channels, and the channels, we plug them here in the membrane to allow the passage. So one of the target responses could be to make facilitated proteins. Are you cold?
I'm okay.
You sure?
Yeah.
Open the window.
No, it's okay.
Okay. All right. Okay. Are we clear? Any questions? Any questions?
I'm like, I get it. It makes sense.
You get it. Thank God. Thank God for the blessing of understanding. There is nothing in the world like understanding. Okay, now, that's an example of membrane proteins. Again, we talked about something else related to membrane proteins. We talked about something else related to membrane proteins. We said that not all proteins who are transmembrane, they are receptors. We have protein channels. We have protein channels. We have protein channels. There are two types of protein channels. Protein channels. We have two types. We have two types of protein channels. These protein channels, we have some of them that are leak channels, and we also have proteins that are leak channels, and we also have gated channels. Now, these protein channels, some of them are leak channel, meaning what? They are like the gate in the park. They are always open. 4 a.m. they are open. 3 a.m. they are open. 5 a.m. they are open. People go smoke weed there. It's open all the time. So now leak channels, they allow the passage of potassium. But be careful, do not assume that one leak channel will allow the passage of multiple different types of ions. That's impossible. So there is one leak channel for potassium, and only potassium can leak through it. One leak channel for potassium and one potassium channel. So one leak channel for potassium and one leak channel for potassium actually goes outside. The leak channel for potassium will go outside. If you go outside, what is that called? Efflux. Efflux. And if you go inside, what is that called? Influx. This potassium that's going to go inside. So potassium goes outside, efflux, and sodium goes inside, influx. Influx of sodium. So now, these channels are open all the time. But we have other types of channels that are closed. These channels are closed. Okay. These channels are closed. These are known as gated. Okay, guys, this is a gated channel. This gated channel is closed, but every gate must have a what? Every gate must have a key that opens it. Do all gated channels have the same key? Do all gated channels have the same key?
I don't think so.
So when they open, some channels, they need a ligand. They need a ligand. These are known as ligand-gated channels. Ligand or stimulus. These are ligand-gated channels. These are ligand-gated channels. You bind from outside, and then they will open, and all of the ions are going to influx inwardly. So you have to have a ligand like acetylcholine, acetylcholine. It's very important for you to remember that. Acetylcholine is a ligand that binds outside. That means it cannot get inside. When it binds, it opens the channel, and there will be an influx of ions intercellularly. There are other gated channels that do not open because of a ligand. They are not open because of a ligand. They are open because of what? Heat, thermal stimuli. When it's so hot, they open. That's how you feel the heat, because these are known as thermoreceptors. So these are thermal dependent gated channels. Some of them, they don't open because of heat. They open because of mechanical stretch, touch. That's how you feel the touch. When you touch them, you stretch them, they're open. Others are voltage gated. So we have voltage gated channel. You have ligand gated. You have thermal voltage. You have also mechanical gated channels. So they open based on a particular stimulus. So it's not just the ligand. It depends on the different kinds of stimulus that lead to the opening of the channel. Okay? So you must remember that. What makes the gated channel open? The ligand, number one. What else? Heat and mechanical stimulation and voltage. Very good. That's what makes them open. So now we have seen, and also this cell membrane. What is green? Always remember to. It was one of the questions on your exam study guides. One of the questions was that those green cubes trying to get into the cell. What's that? That's glucose. Glucose trying to get inside. That's a concentration gradient. But when they had sodium with a positive charge, that's not just concentration, that's an electrochemical. Because sodium is a chemical, but it has a charge, it's positive. Chloride is a chemical. Remember, chloride has a neutron, proton inside, has a nucleus, it has electrons. So it's a chemical, but it's not neutral. It has a charge, so that's the electricity. Now, this is not charged. I can touch it. Okay? But if it's charged, it's going to electrocute me. So that slide, that on the study guide, did not mean it was a concentration gradient. It was a voltage. That was a voltage gradient or electrochemical. 2 in 1. Chemical who's charged. Electrochemical. So we said that these are proteins. We have what types of proteins do we have on a cell membrane? Either transmembrane proteins or integral proteins or peripheral proteins. Then what kind of sugars do you have on a cell membrane? What kind of sugar do you have on a cell membrane? I'm sorry? What kind of sugar do we have on a cell membrane? We have sugars that are attached to the protein. It could be sugar attached to the... What kind of proteins we have? We can either have a peripheral protein, or we can have integral proteins. Okay? Then what happens if you have a sugar who's attached to the peripheral protein or attached to the integral protein? In the end, it's a sugar with a protein that's known as glycoprotein. Thank you. Glycoprotein. Are these the only greens we have? If you're green, you're only attached to a peripheral protein or an integral protein. Is that true? Then where else can you be? So if you're a sugar, you can only be on the peripheral protein or the integral protein. There's nowhere else you can be. Where else can you be besides on a peripheral protein or an integral protein or a transmembrane protein? Where else can you be? What about the lipid? Can you have this? Can you have green on the lipid? Can you have green on the lipid? Yes, yes. What is that called now? That's the glycolipid. Excellent. Glycolipid. And the set of glycolipids, the combination. So on your skin, the sugars on your proteins, the sugars on your lipids, are they the same between everybody? Are they the same? That's like your fingerprint. That's how your immune system is able to detect self from non-self. That's how your immune system is able to do immunosurveillance and say, oh, that's not my body, because that's a different set of, that's called glycocalyx. 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 So we don't have to say, in glycocalyx, we don't have to say glycolipids and glycoprotein. They combine them into one word. What is glycocalyx? That is glycolipids and glycoproteins. So the set, the combination set on your skin and on your cells, every cell has glycocalyx. So my glycocalyx is different from your glycocalyx. That's how when you do blood transfusion, when you do liver transplant, your immune system is going to start screening the surface of these cells. And they say, wow, that's not my glycocalyx. That's an invader. That's an infectious microbe. That's a parasite. That's a bacteria. That's a virus that is going to mount a dangerous immune response. It doesn't understand. Yes, it's different glycocalyx, but it's a liver from somebody else. It will never understand. So it will destroy your liver. So it's your glycocalyx that is used for immunosurveillance. And it's made up of glycosugars and glycoproteins. Everybody has their own set of glycocalyx. Except who? A mother and her baby? Or twins? It's the twins who are identical. Identical twins, they have the same glycocalyx. Everybody else have different. Mommy and her baby have different glycocalyx. Twins who are fraternal have different glycocalyx. It is only twins who are identical. These are the ones who are have a different glycocalyx. Yes. The same, yeah. They have the same. It's interesting because they are identical. What's the difference between an identical twin and fraternal twin? Okay. I'm sorry?
They're on the same side. If they go down the same.
Okay. You have an egg. That's an egg. That's an egg. An identical twin, two sperms, they fertilize the same egg, and the egg divides to give you identical twins. Identical twins. Identical twins. But for fraternal twins, For fraternal twins, you have two different eggs. Two different eggs. Each is fertilized with one separate sperm. And that's how you get fraternal twins. So fraternal twins, they have different placenta, but identical twins, they share everything. They share everything. The placenta, they share everything. Because they come from the same egg. And that's why they have the same glycocalyx. Okay? 5 more minutes and then we'll go on break. Does anybody have a question? Does everybody have a question? Anybody has a question about the cell membrane and the glycocalyx and the different types of proteins we find? Okay. So, we talked about the lipids, we talked about the proteins, and the last thing I want to talk about before we go on break is the sugars. You gotta know your sugars. You have got to know your sugars. These are your last kind of your macromolecules before we go into the fourth one, the nucleic acids today when we come back from break. Very important. Okay, folks. We said that we definitely have to have macromolecules, and we begin with micromolecules. So let's work our way up from micromolecules up to macromolecules. From micromolecules up to macromolecules. So, sugars begin as monomers, known as monosaccharides. Monosaccharides. These monosaccharides are glucose, and thank you, fructose, and lastly, galactose. These are your monomers, these are your monosaccharides, and these are your building blocks. If I connect them 2 in 1, I can have one of 3. I can have obviously disaccharides, and the disaccharides, there's 2 of them. The disaccharides, there's 3 of them actually. You can either be maltose, that's the first one, and maltose is made up of 2 glucoses. Thank you. Glucose and glucose. Connected by a, what's the name of this bond? This oxygen bond in the middle, what's that called? What's the name of this bond? The bond between the 2 amino acids was known as a peptide bond. What's the name of the 2 bonds between the monosaccharide that give us a double disaccharide? What's it called? What's the name of that bond? Nope. Do you see hydrogen there? Not an ionic bond. No, we're not talking about what type. What's the name of it? It's a covalent bond. But what's the name of it? I'm sorry? What is the name of it? That's the glycosidic bond. Maltose is glucose and glucose. While lactose is, what is lactose? Glucose and galactose. And lastly, you have sucrose glucose and glucose and fructose. You get it? You guys get it?
Yeah.
Now, do we only have monosaccharides and monomers, disaccharides, dimers? That's all we have? No, we have polysaccharides. Polysaccharides known as polymers. Who are my three polysaccharides? Excellent. Starch, glycogen, and cellulose. Starch, glycogen, and cellulose. These are my polysaccharides. Okay, if I'm going up from micromolecules, if I'm heading upward from micromolecules up to macromolecules, I'm building up. If I am building up, if I am going up, building up, is that anabolism or catabolism? Anabolism. And if I am building up, am I storing energy or breaking down energy? Somebody break down? If I'm building up micro into macro, from monosaccharide to disaccharide to polysaccharide, I'm storing, so I'm storing energy. I'm storing energy. And when I'm storing energy, am I getting exergonic or endergonic? Endergonic. It's not endo, it's ender, A with the E, endergonic. Is that positive delta G or negative delta G? How is it positive? It's exergonic, the energy is exiting the bond to me. The energy is, the arrow is pointing from the bond to me. So it's exergonic plus delta G. That's when I'm breaking down. But when I am building up, the energy is going from me to the storage. I'm storing the energy. So it's going from me, stored in the bond. So that is negative delta G. But if I'm breaking down, if I'm going downward, if I'm breaking down, is that anabolism or catabolism? That's catabolism. And when catabolism, is that endergonic or exergonic? And hydrolysis is where? Hydrolysis. Is it in catabolism or anabolism? Hydrolysis. Lysis. I use water. Hydrolysis. So I use water to break down. So I'm catabolizing. Excellent. And when I remove water, then I'm joining together. That's the same thing for sugars. Okay? Let us take a half-hour break and we'll reconvene in a half hour.